It's one of the largest organ in the body which helps you in locomotion, and you can see if you take one muscle bundle, it will have some type of muscle bundle, what we call is that these fascicles. Fascicles are covered by connective tissues, and each fascicle will have multiple muscle fiber which are the functional units of muscle tissue, which is a long syncytial muscle cell with multiple nuclei. And if you look more carefully, these muscle fiber, which is a long syncytial muscle fiber, which is actually is a muscle tissue, will have undifferentiated cells called as satellite cells. Okay? These satellite cells forms during the muscle fiber formation, but they remain differentiated and they remain quiescent, and during the injury or the traumatic effect to the muscle tissue, they undergo proliferation. First, they will undergo migration to the area of the injury, and then they proliferate, and then they fuse with the damaged muscle fiber to restore the actual original capacity of the muscle fiber, so to have functional muscle tissue again. So satellite cells are quite important because they can able to regenerate the injured muscle. So why we want to study? Because with satellite cells, when they undergo regeneration, they receive information from the connective tissue and the neighboring muscle fiber as a niche what we call it as an environment in which they receive certain chemical signals that will help them to regenerate and the active muscle tissue. So by studying satellite cells, we can actually know how we can use them to kind of treat muscular diseases, or in terms of speeding up the repair process of the injured muscles. So this is how normally, when you see the athletics people or sportsmen will have injured muscle due to the continuous progress use of muscles that will lead to activation of satellite cells, and also we can actually hack into their system to make them useful for diseases like muscular dystrophy, etc. So therefore, studying the satellite cells in different model organisms is quite important. So, for example, if you study them in model organisms like Drosophila, will facilitate the understanding the mechanism that control both quiescence and proliferation. So AMPs, adult muscle precursors, are one of the best model systems to study the muscle stem cells in invertebrate models. So when I was doing my PhD, I started to work on this. So I would like to explain this diagram for a non-applied biologist, how muscle tissue differentiation happens in Drosophila. As Drosophila is one of the insect, as you know, they undergo metamorphosis. They have two life cycles. One is larval and one is adult life cycle. So most of the tissues that are required for larva will be eliminated during the pupal stages, and then a new tissue will form during pupal stage, either through histolysis or proliferation, and then they give rise to the adult tissue. So basically, Drosophila has two types of tissue or two sets of tissue. One will be used for larval tissue or larval life cycle. One will be used for adult life cycle. So in muscle tissue, you can see the muscle progenitor forms during the embryonic stage, and there are two sets of cells will form. One is differentiated body bone muscles, which will be used for the locomotion of larva, and at the same time, a non-differentiated quiescent muscle stem cells will actually form along with these differentiated muscles. They remain quiescent and stay inactive till the larval stages. But during larval stages, during the pupal stages, they undergo proliferation and they give rise to whole set of adult muscles. So this is another diagram that explains how the molecular mechanism actually help them to formation of the differentiated muscle and muscle stem cells. If you see the mesoderm band of the Drosophila embryo, there is something called competence domain, which gives rise to these muscle tissue. From the competence domain, you have formation of promuscular cluster due to the asymmetric division and also lateral inhibition effect of the genes like Latilas Q, and also with the help of Notch signaling pathway. You can see the formation of this promuscular cluster, which gives rise to muscle tissue. Finally, due to the help of Notch signaling pathway, one cell will become muscle founder, another cell become fusion competent myoblast. Now, this muscle founder cell is more like a key cell in the muscle fiber formation. So these muscle founder cells undergo asymmetric cell division with the help of inscrutable and Notch, and one cell will become founder, which fuse with FCM, form differentiated muscle fiber. Another cell will remain undifferentiated and quiescent for a long time, which we call it as muscle stem cells. So what is interesting, because if you look at the muscle tissue, they form from the same progenitor. Okay, So both differentiating muscles in the green and the blue color, the stem cells from the same progenitor. So how in one division they accumulate so much different cellular mechanism, molecular signature that have, one is remain undifferentiated quiescent for a long time, one is differentiated performing already the activities of muscle tissue. So this was a question we wanted to address. So what could be the thing that is happening inside the cell at the level of genomics or at the level of transcriptomics? To have in one cell division such a big differentiation, one having differentiation, other one having undifferentiated stage. So, and this is the behavior of AMPs. You can see in the embryonic stage they remain undifferentiated, 6 AMPs per segment. During the larval stages, in the early larval stage, they remain undifferentiated. But by third instance larva, they undergo proliferation and gives rise to the adult muscles, as I showed you in the previous slide. So during early stages of my PhD, I generated a transgenic line that shows actually cellular membrane of eggs. So if you go back to the previous diagram, earlier people used to stay only with a nuclear marker. So they used to think that there are 6 dots sitting, not doing anything till the proliferation stage. So when we just put the membrane mark, you can see they form this remarkable philopodial connection between all the eggs in the embryonic system. So this was something new and exciting for us, and we wanted to explore more. So we have tried to make kind of a light imaging. You can see during the early embryonic stages, the philopodial formation will go up and they form connection with the muscle stem cell in the top, and they also form philopodial connection with the side of the Drosophila. So this is the same transgenic line what I showed you before in the life cell imaging through the embryonic development. And after performing this, we wanted to check what happens rather than crossing USGFP system, which I didn't explain what we call as a binary system in Drosophila, by taking a GAL4 and USGFP. I generated another transgenic line that can express the membrane-driven GFP with a direct promoter that's expressing these muscle stem cells. So in this, GFP was more stronger. So along with these thin philopodial connections, we also could able to see a lot of microphilopodia around the each cell. So we wanted to wonder, like, what's the function of this long philopodial connection and this microphilopodia around these cells, which were thought to be a property of an inactive cell. So inactive cell, undifferentiated and quiescent, what it's doing with all these philopodia. So just see the cellular behavior of these philopodia. And at the same time, there's an article in 2010 that shows even the satellite cells in mammals, they form this kind of a cellular structure between each other that shows that they might be exchanging some information to keep them in quiescent and in undifferentiated state. So with these two, we started to observe the behavior of this philopodial structure, and it seems to be disappearing, which is a quite dynamic philopodial structure in the late embryonic stage, in the early larval stages. But if you go to third instance larva, when they start to proliferate, which is shown by twist, it's a nuclear marker. So one cell has become multiple cell. You can see the philopodial structure around the cells and also between the AMPs have been disappearing. So it shows that they are actually interacting with each other and they're communicating to keep them quiescent. When they start to proliferate, they lose this connection. So there's a strong correlation with that. And then we also wanted to see the microphilopodia, what you see, what they're trying to communicate. When you see a philopodia, they're trying to communicate with other cells. So when you look at the microphilopodia, so we could able to stain this MC gap GFP with a 22C10, which marks the PNS. They are kind of aligning on the long philopodia. But if you do a careful live imaging and the three-dimensional movies like this, you can see the microphilopodia are not aligning with this red structure. Here you have a double study of live imaging. The PNS are in the M-cherry and the muscle cells are in the GFP. So they actually forms thin philopodial long structure align the PNS, but however the microphilopodia are on the surface, they are not actually contacting the PNS or the neural system. So then again we repeated the same experiment with the marker that is expressing in muscles. So here you have M-cherry in the muscles now, and you can see the thin philopodial exactly in the skeletal muscle system. So that shows that, as shown in my first slide, the stem cells require a niche for the surrounding environment to maintain their quiescence and then unproliferated state, so that they can exchange signal with this microphilopodia, so that it can remain quiescent and undifferentiated state. So hopefully, this, yeah, that's okay. So then we also wanted to see what's the actual role of muscles. If you remove the muscles, what happened to the AMPs? Here you can see the wild type embryo with muscles in red and muscle stem cell in green, and staying with a marker that express the apoptotic cells. So apoptotic cells are not actually expressed in the muscle stem cells. But if you kind of destroy all the muscle tissue by genetic expression of an apoptotic marker, you can see most are being destroyed, and cell which marks the apoptosis. So you can see a lot of muscles undergoing apoptosis because of you express apoptotic marker, plus also the muscle stem cells in which you did not do any genetic manipulation also undergoing apoptosis. That shows that they heavily depend on the surrounding skeletal muscle tissue for their survival, for their maintenance of quiescence and to remain unproliferated cells. So if you destroy the muscles, they also will undergo apoptosis and start to express the tunnel cell. So this is the kind of completion of the first part. I'm not going to go for genetic analysis, what we did, the gene network and signaling pathways that require them to proliferation. So the question we are asking you from my PhD time onwards, when you look at these structures, muscles and stem cells, differentiated versus undifferentiated, coming from the same progenitor within cell division, what is happening inside a cell at the transcriptomic level? So we wanted to address this just by simply doing a transcriptome analysis, but I don't know if you have worked with Drosophila, or you can think about even a mouse having a small tissue. It's not to just take this cell and isolate an RNA, right? It will be almost impossible technically. So